Hi everybody, welcome to a special Live with Children here in San Diego. It's the very first interview I'm doing for SLES 2023 and I'm delighted to be joined by the CSO for Myllia, Tilmann Burckstummer, who is going to tell me a bit more about Myllia, what they do, but also Tilmann has a poster, as you can see behind him, which he's presenting tomorrow on Monday the 27th, if I get my dates right, where he's going to talk about that as well. So first of all, Tilmann, it's lovely to see you. I hope you had a safe journey here to San Diego.
Yeah, brilliant. No, it was great to get here, and I'm here with friends, so it's always fun to be at these conferences.
Fantastic. Look, Tilmann, my first question for you is, for people not familiar with Myllia, could you give me an overview of the business and who you are?
Yes, I can. So at Myllia we perform CRISPR screens, and we run CRISPR screens with clients in the industry. And so we couple these CRISPR screens to various readouts, including fitness, flow cytometry, and single-cell RNA sequencing.
Right, okay. And in terms of what you do, what's unique about it?
Well, what's unique about it on the one hand is that we use very sophisticated models. So on the one hand we use, and I'm going to speak to that in a moment, primary human T cells as a source, and those are not easy to handle, not easy to culture, not easy to transfuse. On the other hand, what's quite special is that we couple the CRISPR screening experiment to single-cell RNA sequencing as a readout, right. And that requires a lot of sophistication.
Fantastic. Now, as I mentioned at the beginning, you have a poster that you're presenting officially tomorrow, which is right behind you. So can you tell me through the poster what it's about?
Yeah, brilliant. I can do that. So essentially here we showcase the platform that I just mentioned. So it's really about isolating cells from the blood of healthy donors or patients, right. And we work here in primary human T cells, specifically in CD4 positive T cells. So those are so-called helper T cells, right. And they come in different flavors, and what a lot of people would like to know is, if we perturb certain genes in these cells, how does that shift the balance? How does that shift the balance between regulatory T cells, Th1 cells, Th2 cells, and other Th subtypes? Right. And so when we first developed this, we had to make sure that we can transduce these cells and we can knock out certain marker genes. And here we're showing an example of a knockout that's right. B2M. But then we performed actually a large-scale screen where we perturbed a lot of genes, and when we do so, we characterize the resulting cell populations by single-cell RNA sequencing. And what's very powerful down here is that we can annotate various subsets. Here specifically, we're not only perturbing the cells, but we're actually skewing them. That means we're pushing them towards the Th2 subset, and we can find all these markers of Th2 cells expressed in these subpopulations. And then importantly, the key bit is down here, where we perturb specific genes. Those are not sort of novel, but those are genes that people knew were involved in Th2 formation, and we can show that some of these genes, when perturbed, really keep cells in an activated versus a skewed state, or conversely, sort of push cells towards becoming T helper cells.
Right, fantastic. Well, I know that was a very quick overview of the poster. If people would like more detailed information, a proper presentation of that poster, then obviously they come and see you here at the show. Is poster 1085-B, and you'll be presenting this officially tomorrow.
Monday,
which is Monday the 27th. Obviously, though, that's tomorrow for the poster, but I know you're here for the rest of the show anyway, right until Tuesday?
Correct.
So if people want to get a hold of you, how can they get a hold of you to sort of have a chat about what you do?
Well, on the one hand, you could reach out via email, and the email address is shown down here, or you could just reach out via LinkedIn. Importantly, this has a lot of applications in the space of autoimmunity and T-cell immunology and tumor immunology, so we'd be keen to speak to companies that are active in that space.
Brilliant. Well, look, Tilmann, good luck tomorrow with the poster presentations. I hope you get lots of people coming to talk to you and ask you lots of questions, and obviously also catch up with you on the day you're not presenting as well.
Brilliant.
And
thank you, Rizwan.
Safe journey back to Vienna. So there you go, viewers. If you're here at SLS this week in San Diego, catch up with Tilmann, find out more about what Myllia are doing, listen to his presentation. As I said, you can catch him tomorrow here by the poster, and also catch up with him via LinkedIn or through the email to find out more, and have a meeting with him on Tuesday. So once again, Tilmann, thank you for taking the time out to talk to me today. Good luck with the rest of the event. And viewers, thank you for watching. Until next time, as always, stay well and stay safe. Bye-bye.